Welcome to the OmicsLogic Bioinformatics for Precision Medicine program. Precision medicine is changing the way we understand, diagnose, and treat major life-threatening diseases. This transformation is driven in part by computational technologies, as well as high-throughput molecular data that is being collected from patients, animal models, and large-scale cell line experiments. In the program, we will explore how the various omics data types generated in these studies can be analyzed and integrated into the clinical process to understand the basic biology associated with communicable or infectious, as well as non-communicable or chronic diseases, including diagnostic strategies and treatment selection, development, and repurposing. So in the first session, we'll review again the main topics of the program, introduce each one in greater detail, and provide you with a guide how to use associated resources to continue this journey. After this session, you should have a good sense of bioinformatic applications in precision medicine. Completing the session video and associated resources will give you a great grasp on how data analysis skills and new technologies like next-generation sequencing are used today in this field. You will also see various examples that you can use to focus on oncology, neurodegenerative, and infectious diseases. In this session, we will learn about clinical and molecular data in the context of risk profiling, diagnosis, and therapeutic decisions. We will also go through various next-generation sequencing data types such as genomics, transcriptomics, and metagenomics. Finally, we will learn about the process of how discovery in the lab leads to real-life applications, or how insights from bioinformatics analysis can be translated to the clinic. To understand clinical decisions where bioinformatics is used for precision medicine, we will look at various data sources such as basic information about patients or clinical data like age, sex, ethnicity, and their environment, data that describes disease history and diagnosis, treatments, and comorbidities, and molecular profiling that allows us to perform genotyping based on SNPs in the DNA, identify biomarkers, or look at microbiome composition. Clinical data typically includes data such as demographics, labs, and medical history. The list of collected variables can vary significantly based on each patient. In most cases, this data can come from a variety of sources like electronic health records, medication, insurance, claims, and family history. For the most part, it is divided into patient information, as well as describes their interactions or encounters with the medical provider. Recently, the sources have increasingly digitized and integrated, leading to an expanded number of variables with various relevancy for each case. Some studies are starting to incorporate many of the clinical sources of information with molecular profiling, environmental factors, as well as mobile health and even social media. While some of the data types are more accurate than others, these all have been used to study disease. For example, in the current pandemic, all of these have been used to track disease spread and identify potential at-risk individuals or groups. Pharmaceutical companies are referring to this as real-world data, or data that can be used to evaluate responses to treatment and improve selection of patient candidates for clinical trials. For many diseases, molecular data is increasingly important and relies on complex bioinformatics analyses, like gene expression analyses, commonly found SNPs, metabolism information, and an array of various biomarkers of disease and treatment efficacy. These molecular patterns in association with clinical data can help refine strategies for diagnostics and therapy selection. New discoveries in molecular data lead to a better understanding of complex diseases at the subcellular level and help identify novel drug targets or opportunities for drug repurposing. For the clinician, advanced molecular profiling reveals the underlying biological drivers of disease and can help explain disease symptoms using genomics, proteomics, transcriptomics, epigenomics, and even metagenomics. These molecular markers affect how a patient is diagnosed and lead to a number of improvements: risk prediction or risk stratification, prognosis of outcomes for treatment modalities, such as individual drugs or combinations of drugs, selection of target therapeutics for patients with complex diseases, and continuous monitoring of disease changes over time. One of the great examples of how such treatment has affected cancer is the example of breast cancer. So we see that from 1971 to 2011, the rate of survival has increased from about 50% to over 85%. One of the reasons was because studying cancer, Herceptin was developed as a targeted therapeutic. It's a monoclonal antibody that has been used to treat HER2-positive breast cancer patients since the 1990s. It works by targeting the HER2 receptor, preventing cancer growth. Some breast cancers express too much HER2 protein, triggering the cells to multiply very rapidly. Today, this trend with targeted treatment has increased. Other monoclonal antibodies, such as pertuzumab or Perjeta, have been developed. The targeted therapy has gotten even more advanced. TDM1, Kadicla, an antibody-drug combination, has been approved for use in the treatment of HER2-positive breast cancers. Antibody-drug combinations work like a smart bomb, delivering chemotherapy directly to cancer cells by attaching to their HER2-positive receptors. Targeted therapies have transformed the approach to breast cancers and offer an opportunity to cure even later-stage breast cancers in certain patients. Now, a lot of this is done using molecular profiling. One such test, called Oncotype DX, looks at the expression of 21 different genes in a patient's primary tumor. A tumor's gene expression pattern shows whether or not it will be responsive to chemotherapy or whether endocrine therapy alone, such as tamoxifen, could be a better choice. A recent study showed that patients scoring on the low to mid-risk range could safely skip chemotherapy completely, which would lead to avoiding the hair loss, neuropathy, weight loss, and other side effects that often come with chemotherapy. But this approach has further developed, and today, molecular markers of disease have led to a complete reclassification of breast cancers into several different molecular subtypes. These subtypes continue to evolve, leading to a better treatment of this complex disease, which is the fifth leading cause of cancer-related deaths annually, with more than 265,000 people diagnosed with it in the United States alone every year. Other multi-omic studies help change the way that we can understand other cancers, such as liver cancer or hepatocellular carcinoma. In the 2017 publication in Cell Reports, the Cancer Genome Atlas Research Network published their analysis of integrated multi-omics data that led to a much better understanding of liver cancer risk, such as infection with hepatitis B and C, as well as other factors like alcohol consumption, ethnicity, and other comorbidities. Such comprehensive studies continue to yield results even today. More data is being collected and analyzed by bioinformaticians, leading to an even greater precision and making it possible to improve the way treatment is delivered or even find new treatments that are less harmful and more effective for certain patients. The role of next-generation sequencing in medicine has changed dramatically over the past decade. To understand why and how, let's look at several methods of molecular profiling, explore the process of second-generation high-throughput sequencing, also known as next-generation sequencing or NGS, and see how such data can be analyzed to discover specific clinically relevant data points for a medical procedure. The growth in high-throughput data is largely driven by cheaper, faster, and more efficient sequencing called next-generation sequencing, or simply NGS. From about 100 million in 2001, which is the cost of what it took to sequence the full human genome, to approximately 3 million by 2008, and by 2017, the cost of a sequenced genome was close to $1,000. Just recently, Ultimate Genomics announced its goal to make the full genome sequence available at a revolutionary cost of $100. This places whole genome sequencing at a comparable cost to most tests, like the X-ray. Recall that it's been over 70 years since the structure and function of DNA was discovered. 50 years have been spent since that time sequencing the human genomes. In 2015, the 1000 Genome Project brought a significant number of whole genomes sequenced and available to the research community. 100,000 whole genomes were already available by 2018. And today, there's an ever-increasing number of datasets that are available to help researchers and clinicians understand subcellular processes and the DNA. Omics data, including such that is provided by next-generation sequencing, includes a variety of types. Phenomics refers to high-throughput imaging or characterization of phenotype. Genomics allows us to identify mutations in the genome. So what is the observed sequence compared to the reference? Epigenomics allows us to study epigenetic variation, or variation that is not dependent on the actual sequence of the genome. This includes such methods as DNA methylation. Transcriptomics allows us to understand gene expression, or how much of a certain gene is being expressed in a cell or a tissue. This is done using a variety of platforms. Platforms are instruments that produce digital omics data. So those can be divided into two types: short read sequencers and long read sequencers. And they can be used to sequence either targeted, amplicon, or whole genome or untargeted DNA and RNA sequences. In most cases, for clinical use, we will refer to either whole exome or targeted panel sequencing. Because in most cases, whole genome sequencing just carries too much information to be clinically relevant. The number of annotated SNPs is much lower than the total number of positions in the human genome, which is over 3 billion long. Genomic variation is typically calculated by the proportion of known SNPs present in a particular gene. And this can range from genotyping, where we group patients by their genotype or some special features in their genome. And in some cases, we are going to be looking for pathogenic SNPs. SNPs are single nucleotide polymorphisms, or a single nucleotide position that has been identified as relatively prevalent in the population, or over 1% of individuals. For example, BRCA1 and BRCA2 testing is extensively used to identify women that are at risk of developing breast or ovarian cancer. In fact, 44% of women who inherit a harmful BRCA1 genetic mutation will develop ovarian cancer by the age of 80. To summarize, omics data can be used to understand what is happening within the tissue or a single cell. And typically, those can include a variety of DNA changes, gene expression changes, epigenetic changes, as well as microbiome changes in the individual. When combined with clinical or phenotypic data, some of that information can be clinically relevant. That means that it will be used at different stages of identifying risk, developing a precise diagnosis, or even selecting treatment that is most appropriate and effective for the individual. And that is why next-generation sequencing data is central to precision medicine. Most bioinformatic analyses are going to be considered as preclinical. That means it is based on data generated in lab experiments or retrospective studies. To explore these, let's look at examples of cell lines derived from patient tumors in oncology, animal models of human disease, and explore how these can be used alongside databases on various specific diseases, for example, TCGA in oncology. Then we will discuss how such insights can be validated and what considerations they bring to biobanking and clinical trials. Next-generation sequencing technologies and experiments are already affecting research, hospitals, and are used by pharmaceutical industry to design specific treatments and adapt them to the needs of individual patients. A lot of times, not all of these experiments are possible on real patients. Instead, we can select tumors in different types of patients, produce those tumors as immortalized cell lines, and use those cell lines to understand the basic biology. Unfortunately, that biology cannot be fully studied just using cell lines, because cell lines don't have all of the elements of the system or the whole body that actually functions as a whole. And so in many cases, in oncology, instead of just using cell lines, there are also tumors that could be extracted in full from a patient, grafted into an animal model, and then studied in terms of their interaction with the rest of the body. Ultimately, such experiments can provide better insights into biological factors that are relevant for patients, and different types of tests could be first performed on these models before we actually develop a clinical trial where such treatments or diagnostic procedures are used on actual patients. This is also changing the way that we apply big data analysis techniques or bioinformatics in real-world clinical needs. So typical process is to collect, integrate, and analyze big data coming from a variety of sources and ultimately change the way that this can be used in a big data approach for precision medicine. We can see this oftentimes used when large-scale data is being collected on a particular disease. A good example is the Cancer Genome Atlas. The Cancer Genome Atlas is a work performed over several decades to collect samples with multi-omic data on patients and use their clinical phenotypes to find important associations. The cancer genome data is available through the Genome Data Commons portal that allows us to find multi-omics datasets with clinical information about each sample, which gives a much better understanding of how cancer variation is diagnosed and treated and how successful those treatments can be. These large-scale datasets are continuing to be collected, and today we have the All of Us dataset as well. The All of Us research program, which is led by the National Institutes of Health, is building one of the largest biomedical data resources of its kind. The All of Us Research Hub stores health data from a diverse group of participants from across the United States. Researchers can access All of Us data and tools to conduct studies to help improve our understanding of human health. Now, it also provides us with a much better understanding of what we call healthy, or the variation of healthy individuals that can be used as a reference when we study subgroups of patients or patients that are of different ages, sexes, ethnic backgrounds, and socioeconomic status. In addition, there are projects that are aimed at characterizing the healthy condition and preclinical variation. One such example is the NIH Human Microbiome Project that allows us to look at different types of microbiome perturbation that is associated with different types of diseases. These include depression, autism spectrum disorder, cardiovascular disease, asthma, arthritis, cancer, and diabetes. By understanding the microbiome diversity, composition, and changes associated with different types of phenotypes, we can start seeing the impact of the microbiome on diagnostics, but also treatment, because patients with different microbiome can metabolize treatment at different rates. Now that we have reviewed the rationale for bioinformatics and NGS in biomedical research in the clinic, let's review case studies of clinical research with real data. These include oncology, neurodegenerative diseases like Alzheimer's disease, and infectious diseases like COVID-19, malaria, or Ebola. We can look at how viral genomics and evolution can help us understand the spread of a disease and identify variants of concern in pathogens or genetic predisposition of some populations to be more susceptible to acquire disease and potentially have worse outcomes. We can look at immune responses, where we characterize gene expression and study biomarkers or genes that are associated with a particular response. And sometimes we can look at microbiome and see how microbiome plays a role in disease progression, drug metabolism, and also predisposes certain people to different clinical risks. On the OmicsLogic portal, you will find a number of case studies and curated datasets around these different topics. Some of them are based on full analysis of datasets that are already published. Simply by following the steps described in these case studies, you will be able to make important discoveries that today are being used in the clinic. Some of them have to do with drug metabolism, identification of somatic mutations in tumor development, inherited or germline mutations like the sickle cell anemia, rare genetic diseases, as well as drug resistance associated with different types of antibiotics and pathogens. Oftentimes, just by reading the abstract, you get a good grasp of how bioinformaticians can use such data to model precision medicine. For example, on this project, modeling precision medicine of breast cancer, you will find how researchers use machine learning to study transcriptomic data and identify subgroups of individuals that are going to respond differently to different treatments. In the study, researchers use multi-omics data integration to look at different types of molecular profiles, integrate them, and compare them to reference databases like the TCGA to understand response to treatment to drugs that are either already used in the clinic or being studied in clinical trials. To continue learning, we invite you to explore these associated resources for the lecture. We will go over the coursework, the case studies, and other examples from literature. To benefit from today's lecture, you can select one of these to refresh your memory of basic terminology, learn more about the various techniques, or practice what we have learned with curated datasets on the topics that we covered in today's session. The contents of the session correspond with practical lessons that you can complete independently that are available on the OmicsLogic Learn portal. With the full course, specific lessons that are of relevance to today's session include Lesson 2, Bioinformatics in Healthcare from Course 1, Introduction to Bioinformatics. In this lesson, you will get an overview of examples from healthcare area where bioinformatics can be applied. You will also see examples and videos of medical professionals discussing bioinformatics and precision medicine applications. Another good course to look at is Bytes and Molecules. This course is a great place for those who was confused by a lot of the terms that we used in today's lecture. Anyone that would like to review the basic terminology and get a good overview of topics that we cover in this course should look at this course in full. But specifically, we looked at this lesson 6, called Introduction to Genomics. If you are interested in genomics, you can also look at these additional lessons in the course 3 course called Genomics. In lesson 2, you will see how you can work with DNA sequences in R, and in lesson 3, you will get an introduction to large-scale genomics. The full course contains a lot more information that you can choose to explore independently. Some of these will be covered in the future lectures, but do not worry about completing them for now. At the end, you will also find examples that I already mentioned. These examples include such projects like Ebola, immune response changes in cancer, modeling cancer precision medicine, or tuberculosis infection and treatment finding drug resistance variants. So let's recap what we have learned so far. First, we learned how combining clinical and molecular data helps reveal underlying factors contributing to the disease, which further helps us provide a precise diagnosis and effective treatment to the patient. Further, we learned about next-generation sequencing technologies for genomic, transcriptomic, and metagenomic data that provide important insights for precision medicine applications. We also discussed the role of bioinformatics in taking research results from the lab to the clinic to benefit patient treatment and diagnosis. Finally, we introduced a set of case studies in precision medicine, from inherited sickle cell anemia to drug resistance in Mycobacterium tuberculosis. In the next session, session 2 on genomic data analysis, we will go one step further to better understand what is genomic data, how do you perform variant calling analysis, what is the difference between germline and somatic mutations, and how to determine a variant's clinical relevance. At the end of session 2, you will have a thorough understanding of genomic data, variant calling analysis, as well as specific techniques for clinical annotation of variants in cancer.